Hi all, welcome back to Microbiology Lectures. Today we're going to continue talking about viruses, and today we're going to concentrate on talking about the structure of viruses. So what are they made of, and what shapes do they take, and then the nomenclature of viruses, like how do we name viruses. Okay, so let's jump into it. So the structure is pretty basic, as you can imagine, from something that is non-living. Even living things like bacteria and archaea are fairly basic when we look at the different structures that they have. Viruses, being non-living, really are composed of two main units, and they have a viral particle, or a viron, which is going to be the nucleic acid. So that can be RNA or DNA, and we'll talk about that here in a second. And then there's a capsid that surrounds that. Now, that doesn't mean that there are not other units that viruses can have. They can have like a lipid layer. They can have projections, which we call spikes. They could be a bacteriophage, which then would have tails. So there are, but many other things that can be part of it. But in general, the viral structure is these two pieces: the viral particle, or the viron, which is the nucleic acid, surrounded by a capsid. Okay? Now, when we're looking at that nucleic acid, we've talked about this before, that nucleic acid can either be DNA or RNA. Now, the interesting facts, or interesting piece of that, is that the DNA could be circular, like a prokaryotic DNA strand, or it can be linear, like eukaryotic. The RNA can be single-stranded, or it can be double-stranded. And so to denote that, normally you'll see that it's an ssRNA or a dsRNA, so single-stranded RNA or double-stranded RNA. That kind of denotes what kind of RNA they have. So we combine words so to indicate like a region. The capsid plus the nucleic acid is called a nucleocapsid, and the capsid with all the other subunits is normally called a capsomer. And so we'll look at these things as we progress. Another thing that can have a really important effect on the virus structure is whether or not it's enveloped, whether or not it has a lipid membrane-like envelope that goes around it. Okay? So both non-lipid or non-enveloped and enveloped viruses will often have spikes. These spikes are used to enter a cell, so they have corresponding spikes to the receptors of the species that they're going to inhabit or take advantage of. Okay, so the non-enveloped will not have the fatty lipid base like layer around it, and instead it'll just be proteins with spikes, and then the nucleic acid will be hidden inside. If it's enveloped, then it's going to have some kind of fat-like envelope that goes around it. It's based on lipids, spikes, potentially, and then you're going to have that nucleocapsid, which would have the nucleic acid and a protein coat. This is important to kind of denote, because how we deal with the different viruses depends on whether they are enveloped or non-enveloped. So again, spikes are attached to the receptor sites on the host cell. Normally, they correspond to different hosts, and so sometimes viruses are very species specific. Their receptors only work on certain species. Other viruses, their receptors are generalized, and they can enter a lot of different species. It just depends on the type of virus. And so that's what makes some of the viruses pretty dangerous. So like the flu virus, it has the capability of often jumping species. So it can affect birds, and it has the spikes that would allow it to enter bird cells. It can affect pigs, and so it has the receptors to enter pigs, and because pigs and humans have a lot of the similar receptors, then it can jump species to humans. And so different viruses have this capability, and normally we call that zoonotic, the ability to move from one species of animal to the next. Okay? Again, like if they're enveloped, they're going to have a lipid bilayer, or at least a lipid-based layer that goes that creates that envelope covering. And then non-envelope viruses, we often say they're more resistant to disinfectants because most of our disinfectants are alcohol-based. And so an alcohol-based disinfectant, it works by removing fats or breaking down lipids. Okay, so viral shapes, there are 3 general viral shapes, although there is some deviation from these, and sometimes there's a little bit of difference that can be somewhat combined. But for the most part, the 3 shapes are isohedral, which is going to be kind of like 20 flat triangles that form this very unique isohedral shape. Again, remember, these are just proteins, so these are amino acids that are just chained together in kind of that triangular form. The helical is normally an alpha helix. There can be different ones, but it's normally kind of a shape of protein that is very similar, that Linus Pauling described a long time ago, called the alpha helix. And so they're going to have that helical shape. This is like tobacco mosaic virus we talked about a couple lectures ago. And then there's complex viruses. And the complex viruses, we often think of them as like they have a head region and maybe a tail region or a body-like region. That head region could be isohedral, and the tail region could be helical. So it's like the combination of these 2 other shapes together in one entity, one pathogenic or non-living virus. And then off of that, you can have other things. Of course, you can have spikes that are coming off of these. You can have tails or spikes that are coming off the tails that help with attachment and the ability for the virus to enter a cell. Taxonomy is messy. From an evolutionary ecologist perspective, my perspective, it's really messy. And part of that really has to do with the way we name viruses. I think not everybody is on board, even though we have an ICTV committee that publishes classification and how we should name viruses. I'm not so sure we follow the rules, and it's not as clear as it can be. That being said, it's getting much better because now a lot of the viral, what we call viral species, although some of us have a hard time calling them species. Species indicate that they're alive, but the viral species, they're categorized based on their genomes, based on the genetics that they carry. So what is the gene or genetic material that they're carrying, DNA or RNA. And so we're starting to get better at kind of basing our classifications on that versus just basing our classifications on the shape or what type of spikes they have, that kind of stuff. Previously, they were classified based on that, right? The host that they infect, the shape. Still, some are useful. Those classifications can be useful, but I think we're starting to move away from that, and we're moving into describing them based on their genome, or the genomic structure, I guess you could say. So prior to really 2019, there were 8 principal ranks for viral taxonomy, and now there are 15 ranks. And for those of you who are familiar with kind of living organisms taxonomy, it's going to look very similar. So instead of domains, we replace domains when we're talking viral taxonomy with realm. But other than that, pretty much everything else is the same as classifying a living creature, which some people have issues with. Anyhow, we'll talk a little bit more about that, but that's kind of I don't want to get into the weeds too much when it comes to that kind of classification. The interesting or common pieces of classification, probably to denote the differences between living organisms and non-living entities that are classified in the same way. The taxonomic names of viruses are written in italics all the way down to genus. The genus and species of the virus is not italicized. And that's the opposite of living organisms. So living organisms above genera is not in italics, and the genus and species are in italics. Okay? And you'll see that virus families end in Virididae, so that indicates the family is a virus. DAE, most of you will know, that indicates that it's a family, but the virid, or the viri part, is indicating that it's a virus. Normally when we talk about viruses, we call them by their informal or common name. Normally that common name is really indicating what route they infect, so they can be grouped into respiratory viruses, or they can be grouped into viruses that cause a certain disease, and they can be named based on that. So for example, like Eastern equine, Western equine, Venezuelan equine encephalitis. So sometimes they're just called the encephalitis viruses, although they've been separated now, many of them, out into different families. There you can see Virididae, so Toga Virididae and Flavod Virididae. And these guys are separated based on their shape. Okay, so based on the shape that they are and based on the genetic material that they carry. Another thing that I want you to take away from the virus piece of this is often we're interested in the virus, of course, but we're interested often in what's its vector. And so what's the species that can carry it, the vector species that can carry it. Because remember, viruses alone can't do anything. So what can they affect? Do they affect humans? Can they affect human cells? That's important. And then what can carry it? That's the other piece. So the vector and then what organism does it affect is fairly important. Okay? And a lot of the viruses that we know and that we're really worried about in the world are zoonotic viruses. And so zoonotic meaning that it goes animal to human. Yes, I know humans are animals, but it goes animal to human, and a lot of these are called arboviruses. Arbo meaning arthropod, arthropod-borne or arthropod-carried virus. Okay, and there's really three kind of classifications. You have what we call epizootic vectors or epizootic organisms, or I should say epizootic viruses. These are ones that occur in our domestic animals and can be transferred from domestic animals to humans and back. A lot of times the domestic animals, pigs, horses, etc., can amplify the virus and maybe show very little signs of an infection, but then when it gets into humans, it can be detrimental to the individual. Okay, so the epizootic is referring to domestic animals. The enzootic refers to vertebrate hosts that are not domestic animals typically. So these are wild animals, wild populations, and again, it's still based on a vector. So most of the vectors are biting insects or biting invertebrates. So mosquitoes, ticks, fleas, these kind of things are going to pass those viruses on between species, okay, from animal vector or animal host to human host. Okay? Often the enzootic are what we call either spillover, meaning that we're not the definitive host. We just happen to be in the region of these wild animals and we pick them up. And so dengue fever, yellow fever, kind of those kind of things, that virus is normally affecting wild animals, and then when we establish ourselves all over the world, we pick these viruses up because we're encroaching on wild organisms' territory. Then you have a series of viruses that are known to humans, and they happen to stay within the human population and just rotate between different humans, and we call those pediodomestic, and that just means that we have a vector that can just circulate this between individuals, humans, and sometimes they're called urban epidemic cycles, but it's pediodomestic vectors, which just tend to circulate between human populations, within human populations. And some of those species, like dengue fever and yellow fever, are now so ingrained in human populations that you don't need a different vertebrate host, and it can just be passed from human to human over and over and over again in urban regions or populated regions, not even urban environments. All right, next time we're going to talk about bacteriophages and go through kind of how their cycles work and how they affect different bacteria.